Hello friends, in the last class we learned about the important characteristics and classification of subdivision Deuteromycotina. So in the present lecture we are going to learn about the important characteristics of class Hyphomycetes, which is one of the form class under the subdivision Deuteromycotina. So this classification already I explained in my previous lecture. Anyhow, I will cover once again that subdivision Deuteromycotina is majorly divided into three form classes, that is Blastomycetes, Hyphomycetes and Coelomycetes, and Hyphomycetes again further divided into four form orders, that is moniliales, which is also known as Hyphomycetales or hyphales, and second one is the tuberculariales, and third one is the stilbillales, and fourth one agenomycetales. So moniliales again divided into two form families, one is the moniliaceae and dematiaceae. In the moniliales, conidiophores are free in nature, that is they are not united, whereas in case of tuberculariales and stilbillales we can see that conidiophores are united into fascicles. Under tuberculariales one family is there, tuberculariaceae, and under stilbillales one family is there, that is stilbillaceae, and another fourth form order is the agenomycetales. So we will discuss each one by one with important characteristics and what are the genera included under each family. So coming to the important characteristics of the class Hyphomycetes are here conidia and conidiophores, they are born directly on the hyphae, and conidiophores bearing conidia they used to be separate or in aggregate. So that is already I told, conidiophores may be used to be free in nature or they may be united, and they generally conidiophores are arising from the mycelium. And there are certain fungi which lack the conidia that we will discuss in the later slides, and they forming the mycelial structures. Such as mycelial structures means we can understand that these are the sclerotial bodies. So sclerotial bodies, regarding this sclerotia I explained in the my initial classes, that is introduction for this mycology. In the initial classes I explained about these things, so just you go and refer those classes. And these members are identified based on morphology of the conidia, that is how this conidia is looking like, and what is the color of that conidia, whether it is hyaline in color or whether it is brown in color, what is the shape of the conidia. So different categories are there, and majorly this Hyphomycetes is divided into four form orders, that is Hyphomycetales, tuberculariales, stilbillales and agenomycetales. So coming to the first one, Hyphomycetales. In Hyphomycetales, which is also known as moniliales, here the conidiophores and conidia and mycelium they used to be hyaline in color. They are directly produced from the hyaline hyphae, that is mycelium used to be very light in color, and conidiophore and conidia are also light in color, not dark in color. And this moniliaceae is divided into two families, one is the moniliaceae and second one is the dematiaceae. Under family moniliaceae they used to produce free conidiophores or conidiogenous cells from the somatic hyphae, and mycelium, conidiophores and conidia they are hyaline or light colored but not brown or black color. So in moniliaceae what is the main feature means conidiophores are produced from the somatic hyphae and they are free in nature and they are light in color, they are not fused. So what are the different genus included under the family moniliaceae? This is the Ainsworth classification what I am explaining here. So these different genus include like Aspergillus, Penicillium, Pyrrhucularia and Trichoderma, Botrytis and Verticillium. So each one we will discuss what are the important characteristics of each genera. Coming to the Aspergillus. Aspergillus we are very known about this. Generally it is a well known saprophyte and grows on all types of substrates, and it is a weak parasite. It used to grow very easily. Generally what we used to call Aspergillus is a weed of the laboratory. Just remember this word, what is the weed of the laboratory means, it is a Aspergillus. Coming to the mycelium, how it will look like. Mycelium is a septate, that is septation used to be there, and branched mycelium with multinucleate cell, that is single cell consisting of the more than one nuclei, that is multinucleate cells, that is cenocytic in nature, but here septate and branched. And coming to the conidiophore, here you can see in the picture conidiophore. This conidiophore arises from the hyphal cell. So from which hyphal cell this conidiophore arising, that hyphal cell is known as foot cell. So they are arised in singly on somatic hyphae, and these conidiophores are long, they are erect, and here non-septate. Here you can see there is no septation in the conidiophore, there is no septation. And these conidiophores bear at its tips like a structure. Here one vesicle is there, and above the vesicle some structures are there like sterigmata or phyalites. These sterigmata or phyalites directly they will bear the conidia. That conidia used to be produced in chains. If conidia are produced in the chains, then we call it as a catenate conidia. That is, catenate means a chains of conidia. And here two layers of sterigmata used to be there. So in the first layer, what we used to call, that is lowermost, is called the primary sterigmata, and upper one is the secondary sterigmata. Coming to the conidia, how this conidia look like in the Aspergillus. Conidia is globose, that is round in shape, and it is a one-celled. Here you can see small conidia, and multinucleate, and it is a thick, rough walled, and used to be, this conidia used to be black in color. Example for the Aspergillus species are the Aspergillus niger, which used to cause collar rot of groundnut. In groundnut, it used to cause collar rot disease. And coming to the second genus, that is Penicillium. The name itself, Penicillium, means it is looking like a broom like structure, that is penicillus. That's why the genus name it came Penicillium. So conidial operators technically is called here penicillus, because it resembles a small brush or broom, hence the name Penicillium. How the mycelium look like in the Penicillium means it is highly branched and septate in nature, and conidiophores, so they arise from any cell of the hyphae, not specifically from the foot cell as we observed in the Aspergillus. Here conidiophores arise from any cell of the hyphae and branch once or twice about 2/3 of the way to the tip in a characteristic either symmetric or asymmetric broom like fashion. So the first generation of branches, that is primary branches, are generally known as rami, and the secondary generation of branches are called metulae. And each metulae ultimately it will produce the bottle shaped phyalites, and on these phyalites conidia used to be there. And conidia are globose and hyaline in color. Here you can see that these primary branches are known as rami, and secondary branches are known as metulae. And here conidiophore, the septation used to be there, and here phyalites are there. On above phyalites conidia are produced, that is globose conidia, hyaline produced in the chains. Coming to the third genus, that is Pyrrhucularia. In Pyrrhucularia, we are very famous about this because it used to cause disease paddy blast, that is Pyrrhucularia oryzae. In Pyrrhucularia, how the mycelium look like means mycelium is a branched septate, hyaline in color and lightly colored, and conidiophores generally they used to emerge from the stomata or through epidermal cells and cuticle, because after rupturing of the epidermis these conidiophores emerge out. And this conidiophore is a simple, erect and septate. They used to be hyaline or light colored with few or several denticulate spore bearing pecs at the tips. Denticulate spore bearing pecs means it is some pecs used to be there on which these spores used to be produced. Here you can see in the picture denticulate pecs like structures on which spores used to be produced. And conidia, coming to the conidia, here you can see the conidia, they are hyaline in color and they are multi-celled, that is 3-celled and broader at the base and tapering towards the apex. Based on this structure, here you can see it is a 3-celled and broader at the bottom, and apex is to be tapered. Example, Pyroclaria oryzae, which used to cause the paddy blast disease. Coming to the fourth genus, Trichoderma. In Trichoderma, these members are generally saprophytes in nature and they are found in soil, and several species we will utilize them as a antagonistic because they used to produce non-volatile antibiotics against a range of plant pathogens. So Trichoderma we will utilize it as a biocontrol agent. These are easily recognized by rapidly growing white, yellow or green colonies. Trichoderma used to be different colors, colonies. They used to form white colonies, yellow colonies or green colonies. Coming to the conidiophore, here in this picture you can see how it is looking like. Conidiophores are hyaline, erect, are solitary, or they aggregated into tufts. Either they used to be single or they used to be aggregated. And here heavy branching we can see in the Trichoderma. This here you can see the groups of conidia also on the Trichoderma conidiophores. Conidia also hyaline grey colored, they are one celled. Here you can see slimy moles, and this ovoid in shape and borne in small terminal clusters as balls on phyalites. These conidia are produced in a ball like structures, many conidia, which we can see on the phyalites. Examples for the Trichoderma are Trichoderma viridi and Trichoderma hargianum. We can utilize them as a biocontrol. Coming to the next one, botrytis. In botrytis, generally botrytis used to cause grey mold diseases in various crops. In botrytis conidiophores, they are branched septate and they used to be long, slender and hyaline in color. Here in this picture you can see how this botrytis is looking like. It is like a grapes bunch. So, and if you see the conidia, the apical cell of conidiophore with the swollen tips, they are bearing clusters of conidia on short sterigmata. Sterigmata is very short in nature. On that sterigmata, conidia used to be produced, and conidia are also hyaline and single celled only here. Entire structure resembles like a grape bunch. Example, the botrytis cinerea, which used to cause grey mold of gram and bean, apple and grape. Here you can see the grey mold disease. Coming to the next one, that is verticillium. In verticillium, here conidiophore, you can see in the picture how it is looking like. It is very slender and it is a septate, and again branching pattern we can see in the conidiophores, and some of the branches bearing verticillate phyalites that give rise to conidia. Verticillate means in whorls, in different holes it used to be produced. So this very slender conidiophore we can observe in the verticillium, and conidia is ovoid to ellipsoid and it is a single cell, and conidia are produced either in singly or in moist clusters apically, like a mucus substance they used to produce in the clusters, conidia attached together in the mucus. Example for the verticillium are the Verticillium alboatrum, which used to cause wilt of cotton and tomato. Generally verticillium used to cause wilt disease in various crops. Then Verticillium dahliae, which used to cause wilt of tobacco and brinjal. And coming to the another family, Dematiaceae. One is under monilials we are studying. So second, Dematiaceae. Here Dematiaceae, this hyphae, conidiophore and conidia, they are brown or black in color, that is dark color, whereas in moniliaceae they are hyaline in color, that is light colored. In Dematiaceae they are brown or black color, but sometimes the hyphae alone or the conidia only dark. It doesn't mean that they should be, all the structures should be dark in color, that is mycelium, conidiophore, conidia, all should be dark in color. It doesn't like that. So sometimes conidia may be dark in color or mycelium may be dark in color, it will vary. So what are the different genera included under the Dematiaceae? That is Alternaria, Drusillera, Helminthosporium, Bipolaris, Cercospora and Physaryapsis. So let us discuss each one. Coming to the first one, Alternaria. Alternaria is also very famous because it used to cause very severe diseases in many of the crops. And coming to the mycelium, mycelium is the in Deuteromycotina we are seeing only the branched mycelium and septate. Here one specific feature is that dark brown in nature. And coming to the conidiophores, they are simple, straight or curved. Here you can see the conidiophore and 1, 2, 3 septate and dark color in nature. And conidia which is a dictyospore, and here you can see brown color. Here in the second picture you can see brown colored conidia, and the shape is obclavate with a beak. Here you can see the beak. This beak size used to be vary among the different Alternaria species. It is not constant character. And 3 to 8 transversely septate and 1 to 2 longitudinally or obliquely septate. That means here you can see the septation. This is longitudinal and transverse septation. Here you can see different septation. So in both directions we can see the septation in the conidia. And conidia are produced acropitally in chains through the pores formed at the apex of the beak of the conidia. So these conidia, how they are produced? Acropital manner. Acropital means oldest at the base and youngest at the top. Here you can see in this picture, youngest at the top and oldest at the bottom, that is acropital manner. Examples are the Alternaria solani, which use cause early blight on tomato and potato, Alternaria brassicae, leaf spot in the crucible. Looking like here they are obclavate, and here this bipolaris, they are germinate characteristically from two polar ends and fusoid and slightly curved. Here slight curvation used to be there in the bipolaris. Examples for the bipolaris are Bipolaris oryzae, that which used to cause brown spot of paddy, and perfect stage is a Cochleobolus myabinus. Old name is, we used to call it as a Dischlera oryzae, but it renamed as a Bipolaris oryzae. And another one is the Cercospora. We are very familiar with the Cercospora, which used to cause Zika disease in groundnut. And coming to the mycelium structure, how this mycelium looking like in the Cercospora, it used to be immersed in host tissue and branched septate and pale brown in color. Here we can observe that it is a pale brown, not dark brown. So conidiophores, they are emerge in clusters through stomata, and conidiophores are brown in color. Septate, simple, are rarely branched with knee joints. The knee joints are very common on the conidiophores, and marking the scars of fallen spores. And how this conidia look like? Conidia are terminal, arise singly from the conidiophores, hyaline, and here filiform shape. Cercospora conidia are how they are looking like? They are looking like filiform in shape, and they are several celled. Here in picture you can see how they are looking like. They are very several celled, that is 4 to 12 septate and a scar at the base. A scar used to be present even in the Cercospora also. Examples are the Cercospora arachidicola, which used to cause early leaf spot on the groundnut. Perfect stage for the Cercospora arachidicola is the Mycospirella arachidicola. And coming to the another genera, Physoriopsis, which is also known as Cercosporidium. Here the mycelium look like with the septate and intercellular, that is mycelium used to spread between the host cells, that is intercellular. Intracellular means it used to penetrate within the cell, but here intercellular with branched haustoria, that it will send the haustoria into the cells to absorb the nutrients from the host tissue, and pale brown in colored and immersely entirely in the leaf tissue. This mycelium, it used to be immersed completely in the leaf tissue, and it used to spread between the cells, and it will send haustoria into the cell to absorb the nutrients from the host tissue. And conidiophores, they are emerging through the ruptured epidermis in clusters, and pale to olivaceous brown in color. Conidiophores are smooth and geniculate, septate, simple with prominent conidial scars. And conidia is light colored. Here you see the differentiation. Under Dematius, it is not compulsory that all conidia look like brown in color. Here light colored and they are cylindrical, usually straight or slightly curved. Here you can see slightly curved, or they are straight in nature, rounded at the base, and shortly tapered with a conspicuous hilum, and mostly 3 to 4 septate. Here you can see on the spores how they are septation. Example for the Hyphomycetes is the Hyphomycetes personata, which used to cause late leaf spot on the groundnut. Cercospora arachidicola causes early leaf spot on the groundnut, and late leaf spot is caused by Hyphomycetes personata. Perfect stage is Mycospirilla berkeley. Mycospirilla arachidicola for early leaf spot. And form order, that is second one is the tubercularials. So first we covered about the form order monilials, that is Hyphomycetes, and coming to the second order tubercularials, it includes the fungi which produce the sporodogium. This sporodogium is a structure where conidiophores are used to merge together. That is, sporodogium is a cushion shaped structure consisting of cluster of conidiophores with conidia. They went together on a mass of hyphae. These conidiophores and conidia, they are produced on this mass of hyphae. This sporodogium is nothing but mass of hyphae. Here you can see the sporodogium structures. Here in the second picture also you can see on which these conidiophores are produced. Under form order tubercularials, only one family is there, that is Tuberculariaceae. Under Tuberculariaceae, two genera are included, Fusarium and Myrothecium. So coming to the Fusarium, the mycelium look like it is a superficial in nature and cottony in culture, and it is septate, hyaline and grouped into a sporodokia. That mycelium grouped into a sporodokia structure. And coming to the conidiophora, this is a slender, short, hyaline and simple, and stout or branched irregularly, bearing a whole of spore producing structures, that is phyalides, bearing the conidia. Coming to the conidia, here we can see in the Fusarium three types of conidia, that is one is macroconidia and one is microconidia, and another one is the chlamydospores. What is macroconidia? Macroconidia, these are the name itself indicating that macro means they are bigger in size. They are several celled and slightly curved or bent or pointed at both the hands. Here you can see the macroconidia, and sickle shaped with a foot cell and hyaline in color. Whereas microconidia, here you can see at the bottom, microconidia are one or two celled, ovoid, and they are single or in chains. They are also hyaline in nature. And coming to the third type of spores, chlamydospores. Chlamydospores are produced from the mycelium. Either they used to produce at the tip or intercalary. They are intercalary in growth, or at the terminal portion they used to produce. These chlamydospores are hyaline, but they are thick walled. That is, these are the resting spores, chlamydospores. Terminal or intercalary, and produced either single chlamydospores, or they used to produce in chains, that is catenate, by the mycelial hyphae or macroconidia. Even macroconidia itself also can capable of producing these chlamydospores. And this how these chlamydospores are formed means they are formed by modification of the previous cell, that is from the macroconidia, or directly from the mycelium these chlamydospores are produced. Examples are the these generally Fusarium verticillium species, they used to cause wilt disease in various crops, and here we will see three examples. Fusarium oxysporum formal species Caesari, that is wilt of gram. Here you can see how it is Fusarium causing wilt in the chickpea, and Fusarium oxysporum formal species vasinfectum, that is wilt of cotton, and Fusarium oxysporum formal species woodum, that is wilt on red gram. And coming to the second genus, that is Myrothecium, sporodokia, looking like cushion like marginal hyaline setae with setae like structures used to be present, and conidiophores are subhyaline to colored in nature, and repeatedly they used to be branched. Here this is the Myrothecium. Here you can see the branching of the conidiophores, and conidia are single celled subhyaline to dark ovoid, gathering in slimy masses. Here they used to be gathered in either slimy masses, or they used to be present in a single. Example are the Myrothecium roridum, which used to cause short holes on the tomato and bendi. Here you can see the this short holes means after drying of this portion they used to create a hole like structure on the leaf. So these are the symptoms. And coming to the form order Stilbellaceae, which consisting of the single family Stilbellaceae. Stilbellaceae include the fungi which produce the cinnamata structure. Cinnamata is nothing but it is a structure in which conidiophores are united together. Here you can see the in picture where conidiophores are united together throughout their length but free at their tip, and they are producing slimy head of conidia at their tip all around the aggregated conidiophores. So they may produce conidia at the tip or throughout the aggregation of this conidiophore they may produce the conidia. Then whole structure resembles like a long feather duster, or it is looking like a brush like structure. Under this Stilbellaceae, one genus is there, that is Graphium. In Graphium, how the cinnamata look like means it is a tall, dark, bearing a rounded terminal mass of conidia embedded in a mucus. Here it is embedded in a mucus like structure, and conidiophores are simple hyaline produced in abundance bearing the oblong conidia. Example is the Graphium ulmi, which used to cause Dutch elm disease. This disease generally is transmitted by beetles. If you want to do details about this disease, just make a comment in the comment section. And coming to the form order Ainsworth, that is Ainsworth, it includes the fungi which do not produce the conidia. That means the fungi which are included in Ainsworth, they do not produce any conidia. They form the sclerotial bodies, that is modification of mycelial structures, and reproduction generally by random fragmentation of the hyphae. So how the reproduction takes place? Because of the random fragmentation of the hyphae. Here two genera are there, Sclerotium and Rhizoctonia. So let us discuss each one by one. Coming to the Sclerotium spores are lacking, that is spores or conidia are absent. Mycelium look like white or light colored. Even in paddy, if you will see, you can find this white colored nature of this Sclerotium. Sclerotia are very hard and they are dark brown in color. Here you can see in the picture, and these sclerotial bodies used to be round in shape. Oblong sclerotial bodies also we can see, and compact, bigger than sclerotial bodies of Rhizoctonia, whereas in Rhizoctonia we will find smaller sclerotial bodies. Here in Sclerotium, the sclerotia is more than 1 millimeter in diameter and consisting of colorless to light color thin walled rectangular. Here two types of cells are there, that is light color thin walled rectangular cells, that is rectangular cells which are inside the stomata, that is inside the sclerotia, and brown to black thick walled cells at the periphery. Examples of the Sclerotium are Sclerotium rolfsi, root rot of groundnut disease it used to cause, and Sclerotium variae, stem rot of paddy. In stem rot of paddy we can observe the white mycelial growth. And coming to the Rhizoctonia, here there is no spores, and here mycelium you can see how it is growing. It is brown in color, stout and septate. Branches arise at acute angles. Here you can see acute angle branches in these both pictures you can see, and hyphal cells barrel shaped and long. How the sclerotia looking like means it used to be black and variable in form. Sclerotia here there is no particular shape, it may be globose, it may be oval, or it may be irregular, and it is loosely formed and connected by mycelial threads. Here you can see how it is connected. And however sclerotial bodies are hard in nature, but it is small in size, less than 1 millimeter diameter, and there is no differentiation of sclerotial tissues. Examples are the Rhizoctonia bataticola, which used to cause charcoal rot of soybean, and Rhizoctonia solani, black scurf of potato, and even sheath blight of paddy. So today in this lecture what we learned various different form orders under the Hyphomycetes class. So in the next lecture I will explain about the Coelomycetes. So thank you for your keen interest in this video, and remember to like and subscribe for more videos, and also share with friends. If you will subscribe more, then I will get interest to upload more videos. I wish you a great success, health, prosperity and happiness. Thank you one and all.